Thank you. It's an honor to be here, and it's an honor to present the first time to this meeting, Asterias Biotherapeutics. We are a publicly traded company, and we're healthy 22 people in Menlo Park, California. I just want to alert you to our forward-looking statement because we are a publicly traded company. So, Asterias' programs are built on human embryonic stem cell technology. That platform was acquired late last year from Geron Corporation, and it represents over 12 years of research and development that was performed by Geron to develop cell banks, cell lines, and various other therapeutic products, particularly our OPC1 product, that were transferred to Asterias. It also represents considerable amount of documentation, existing INDs, and in fact a considerable IP portfolio that has again been transferred to Asterias. So Asterias has two lead products that really address various large markets with significant unmet medical needs. The first product is referred to as AST-OPC1, and they are oligodendrocyte progenitors that are being dedicated and targeted to the treatment of neurodegenerative diseases. The first indication, the lead indication, is in spinal cord injury, a devastating injury that affects more than 12,000 individuals within the United States and really represents about over $14 billion per year in just costs in taking care of these individuals and maintaining the health care of these individuals. Our line extensions in this we're looking at stroke and in progressive multiple sclerosis. The second product that we're developing is referred to as AST-VAC2. Its lead indications are in oncology, potentially in the future in infectious diseases. But the lead clinical trial and clinical indication that I'll be telling you a little bit about later on is in lung cancer. Again, this is a dendritic cell-based product, and line extensions we're looking at is because of the antigens that we have chosen could be in other cancers. So I'll go through both of these two particular products. The first product, again, AST-OPC1, they're oligodendrocyte progenitor cells that are derived from human embryonic stem cells. They have been in the clinic, and we've shown now that through extensive non-clinical studies that we can get engraftment of these cells over a year. We can get significant injury cavity filling, as demonstrated in the lower left picture here, that we can see improved locomotor activity, and there is no observed toxicity or immunogenicity. We know that these cells work by multiple mechanisms of action. First, we demonstrated that they can remyelinate axons, that they produce neurotrophic factors, and in fact that they can induce revascularization. So through initial study that was started by Geron and patients who are now being followed up for the long term by Asterias, we can show that these cells, when administered to patients, are safe, at least initially in the first clinical trial. The first clinical trial essentially involved the administration of these cells to patients with neurologically complete thoracic spinal cord injuries. And these patients have been followed out now for the earliest one is almost 4 years right now. 5 subjects received a low dose of 2 million cells and followed for 2 to 3 years. The earliest one now, almost 4. We've seen that there is a clean safety profile established with these cells. There's been no serious adverse events that have been related to the surgery to implant them, to the cells themselves, or to the immunosuppression that is co-administered in the early stages of the administration of these cells. There have been no unexpected deteriorations in neurological function, no unexpected improvements in neurological function. There, again, no adverse effects seen on MRI, so there's no evidence of tumor teratoma formation. There's no evidence, again, of deterioration of the tissue of the spinal cord. And we've shown that there are, with regards to immune responses, looking at both the cerebral spinal fluid and, in fact, looking at peripheral blood, that we see no evidence of antibody or cytotoxic T cell responses to these cells. We're seeing some potential indications of activity by MRI. We are seeing that there is reduced cavity formation that is in these individuals, even with this low dose of cells, and we're following up on that particular observation right now. We are now proceeding with a new clinical trial that will be starting in early next year. This has received clearance from the FDA, and it is a trial where we'll be looking at dose escalation, starting with the dose that we started with in thoracic patients, escalating of 2 million cells and escalating up to 20 million cells. The importance of this particular trial now is that we're looking at patients with complete cervical spinal cord injuries, which we believe will be the first target registration population for this particular product. Again, we're looking for safety, we're looking for effects on upper extremity motor function, we're looking for improvements in upper neurological function, and obviously looking at testing out a variety of new efficacy measures, outcome measures that have been now introduced in the spinal cord community. What we're looking at is what this trial could lead to would be a phase 2B expansion trial to verify the efficacy of an activity of this particular product, which eventually would lead on to a phase 3 clinical trial. One of the things that's been a pleasure is that we have partnered with CIRM on this particular program and have received an SP3, a Strategic Partnering 3 award that essentially will provide us about half of the funds to execute not only this particular clinical trial, but also some of the product development activities that will be leading on to improve processes to enable advanced stage clinical trials. The second product that we are looking at are dendritic cells. Dendritic cells derive from embryonic stem cells and that are pulsed with telomerase. Again, the idea, the proof of concept of this is dendritic cells have been shown to be active in a variety of different clinical trials, including one that was established by Geron using autologous dendritic cells. The fact that we're moving on to an allogeneic product derived from embryonic stem cells will provide us an off-the-shelf, on-demand product, significantly reduce the costs of producing this product. And we've shown already in a variety of different studies that they do produce mature dendritic cells that will present tumor antigens. And one of the ideal things that we find associated with this allogeneic cell is that it could produce potentially an adjuvant effect. The allogeneic responses to the actual cells themselves could act as an adjuvant, as a danger signal to promote antigen presentation to the host immune system. Obviously, one of the things that we're looking at right now is the antigen that we will be pulsing these cells with. We are going to continue with the antigen telomerase that we had been using in our autologous trials. Again, the rationale for this comes from the use of autologous dendritic cells that were pulsed with telomerase that Geron had executed in 2 different phase 1 and phase 2 clinical studies in prostate cancer and in AML that essentially showed that you could induce anti-telomerase immune responses and anti-tumor responses when administered to patients with these indications, and that we did see beneficial immune effects and effects on the outcomes of these individuals. So we are going to be proceeding now with a clinical trial in the UK where we will be looking at administering these dendritic cells first at low dose of 10 to the 6 cells per injection, then escalating to 10 to the 7 cells per per injection, and we'll be looking at 2 different patients with lung cancers. The first population, those individuals with resected lung cancer who have no evidence of any metastatic disease. We'll be looking at trying to see if we can get immune responses and the safety of this product. We will also eventually be advancing to patients with advanced lung cancer to see if we can have any particular impact on those individuals, and again, in terms of immune responses and anywhere of disease-free survival or relapse rates. So the results, again, here will provide us the impetus to go for either a large randomized phase 2 clinical trial, looking at combination trials with various checkpoint inhibitors, and looking at other cancer indications. So the one thing that we're very excited about here is that in executing this trial, we have also established a partnership with Cancer Research UK, who essentially will be running this trial and doing all of the manufacturing of the VAC2 product that will be supplied for the particular clinical trial. So it's been an exciting partnership so far. We are transferring the technology as we speak to CRUK and their manufacturing center for the execution of the trial and filing of the IND later on next year. So I think I'll end here, but just to say that we now have focused on Asterias is focused on 2 particular products. Our OPC1 product that is re-entering clinical trials in a population of patients that we believe will represent the target population for registration. We're also actively pursuing our dendritic cell platform with CRUK and looking at initiating those clinical trials in about a year and a quarter. So we're also looking at other products from the platform that we received from Geron, but we're looking at it in a staged manner, looking at advancing our lead products, and then eventually, as time goes, taking on new programs. So thank you.